we were able to take a new functional molecule from that addition of that functional group. We now have a new molecule. So what we're going to do is we're going to talk about 4 main macromolecules. And if you remember which ones they are, we had carbohydrates, we had lipids, we had proteins, and we had our DNA, RNA. These were our and carbohydrates are going to be the first one I'm talking about, of which you're going to be able to see on here. An example of one of our carbohydrates. If you're thinking carbohydrates, yes, these are the same carbohydrates that you see on the labels of food. This is our way of measuring, or our way of knowing that this is a specific carbohydrate. And the way we can know on that structure, typically what you would see are hydroxyl groups attached to a carbon backbone. You would typically see those, and something that you would also see that's characteristic about carbohydrates is that they can form rings. So you get sugars that typically have 6 carbons on them. Before I do that, I want to get into the concept of what a monomer and a polymer. So the term we're looking for on here, the prefix is mono, referring to 1. And polymer, referring to many, to the poly. And so when we look at monomers and polymers, monomer is a single unit. A polymer is a unit with a bunch of those single monomers attached. And when we look at carbohydrates, we have polymers that we'll be talking about. But first we have to start off with our single or monomer for carbohydrates. And these are what are referred to as simple sugars. Simple sugars because it contains 1 saccharide. And the suffix here, saccharide, which you've probably heard the term for saccharin, which is sweet, means 1 sugar, hence the term simple sugars. And these simple sugars, you can see we have 3 types of simple sugars. We have glucose, we have fructose, and we have galactose. Glucose, of which you may have heard of, is something that we use as a readily formed energy source. Fructose is a simple sugar that's found in fruits. Have you ever heard of high fructose corn syrup? That's a bunch of fructose all crammed together. Fructose, if you were to taste glucose, has a sweetness to it. If you tasted fructose, fructose in the same weight is much, much more sweeter. Hence why if you were to look at why we use high fructose corn syrup as a sugar substitute, it's because you can get a lot more bang for your buck. When you can create a bunch of fructose, it tastes as sweet, you can use less of it, but still not good for you. Galactose is the other component in milk sugar that we won't really talk too much about, but it's that 3rd monosaccharide. Now disaccharides, on the other hand, are 2 sugars attached together by a glycosidic bond. You don't need to know the term for that. Glycosidic bond is just a bond that forms those 2 sugars together to create a disaccharide, of which we'll talk about 3. Number 1 is sucrose, which is a combination of glucose and fructose. That forms table sugar. Anytime you buy sugar, that's what it is, glucose and fructose combined together. If you look at lactose, lactose is a milk sugar. That if you read milk and you look at the back label and you think, oh, this is high in sugar. What exactly are those sugars? That's lactose. And lactose is a combination of glucose and galactose. Is anybody here lactose intolerant? Really, that many people? Just 4 of us? Okay, well, lactose intolerance is the inability to break down glucose, or break down lactose into glucose and galactose. And we'll talk about why we have that inability. Maltose is grain sugar. If you hear of malt, like malt liquor or malt ice cream, that's basically the sugars that come off barley. You'll see a lot typically in barley or other grains, you extract the sugars. If you ever think of malt liquor, what they're taking with those maltose is they sit there and they ferment it over time and it turns into some form of ethyl alcohol. And the reason I'm kind of going to more examples of this is because I took a beer brewing class not too long ago to kind of burn 4 hours of time. But learned a lot about it, but never relevant because I actually never made anything. But nonetheless, that's maltose. Glucose plus glucose. Polysaccharides is a string of sugars all put together. And if you think about a whole string of sugars, think of like a Pez container. You have a bunch of glucose molecules strung together. Those are your polysaccharides. It's a polymer of a bunch of carbohydrates. And this is what's referred to as complex carbohydrates. So if you ever look at, again, the back of a food label and you wonder what's the difference between simple sugars and complex sugars or complex carbohydrates, that's exactly what it is. It's a bunch of carbohydrates strung together. And how we store our energy, if you think of why do we need complex carbohydrates, it's how we store energy. We store it in the form of, and I'll talk about this in just a little bit, it's our storage form. And in fact, plants do the same thing. They store a bunch of glucose molecules together. So the first one, let me first refer to as starch. Starch, we know high starchy foods like potatoes and, can't think of any other starchy foods, but other high starchy foods. They contain a bunch of complex carbohydrates. And why? Because they string a bunch of glucose together in starch molecule. So that way, and you'll learn about this in cellular respiration, when that plant needs to burn a glucose molecule, it does it by using that starch. It breaks down that starch, that complex carbohydrate, breaks off one of those glucose molecules so it can undergo cellular respiration. So you would have heard about plants. They undergo photosynthesis and they create glucose. They store glucose, like for potatoes, for instance, they store it in the root. So when there is no sun and the plant continually needs glucose in order to undergo and create energy, it can tap into its starch resources to create energy or to redeem energy from those glucose molecules. Now, we don't form starch. We form our polysaccharides in the form of glycogen. And where do we store that glycogen? We store it in the liver and we store it in the muscle. So you'll learn in cellular respiration, when we have a muscle cell that needs to burn energy, it gets it specifically from the glycogen. It takes that glycogen, and if you were to look at these complex carbohydrates, they're literally strung together. It will just break one off, and now you've got a glucose molecule to use and create energy from it. We're going to undergo an exergonic reaction where we take that glucose molecule, again this glucose molecule, and turn it into energy. So where do we store most of our energy? Again, don't necessarily need to know this per se, but 75% of all glycogen is stored in the muscle, and about 25% stored in the liver. And this will make much more sense when we talk about this on Thursday. You'll see the processes in which we generate glucose. And I'll have a lot of terms for you to review as we go through how we generate glucose and how we burn glucose. The last bit of complex carbohydrates is cellulose. And cellulose is very prominent in cell walls of plants. That's what gives those plants those very strong cellular structures, these walls on the outside that we can't necessarily digest. Other species, other animals can digest these, but we have no use for it. The only use that we use this for is the fiber that's in our diet. So if you ever look again at the back of the label, it says dietary fiber, or it says insoluble fiber. This is fiber that we can't digest, but it comprises most of the roughage that you find in feces. That's the stuff that's left over. So if you ever eat like a bunch of salads, you know you've got bulkier stools, as opposed to if you had a low fiber diet, you probably know what your stool is like. So that is our carbohydrates. One thing I want to mention too, because you saw me drawing our glucose molecules in a ring. What's not, what typically longer molecules like glucose, and let me just draw out that diagram I was drawing, 1, 2, 3, 4, 5, and then one of these. These are carbons attached at each single one of these points. Typically what happens with these long carbohydrate molecules is that they will start to form a ring. This one will attach back to this point over here, and then you get a ring-formed carbohydrate. And when you look at glucose that next Thursday, you'll see that ring form, and we'll talk about how it gets broken down. We won't get into the real molecular breakdown, but we will get to the gist of the conformational changes that occur to break down and release our form of energy from it. So that is our carbohydrates. Let's take a look at lipids. And lipids are important because these are the most diverse form of biomolecules. Why is it so diverse? You think lipids, fats. Fats are bad. No, not really. Fats are not bad. Back in, I think maybe like 90s, early 2000s, we had like the, I think it was not the Atkins diet. We had like low-fat diets. People used to think that fat is really bad for us. And so people eliminated fat from their diets, but they found also there was a whole host of side effects that people were having, this mental dysfunction, not mental dysfunction, but reduced function, like mental function, etc. Why? Because fats are important for us. We have 4 categories that these fall under, that fall under the category of lipids. We have fatty acids, triglycerides, phospholipids, and steroids. I'll talk about these in just a second. What exactly are lipids? Lipids are long strings of, typically with hydrocarbons. And they're comprised of a bunch of carbons and hydrogens. Things you can see, like for instance, on our next slide over, you can see a whole string of carbons, hydrocarbons, etc. These typically in room temperature are solid. Any fats that in room temperature that are solid at room temperature are typically fats. These are like your saturated fats, like butter and like animal fats. Anything that are liquid at room temperature is typically oils. So you see fats and oils. There's plant oils, there's fish oils, you name it. Another thing about lipids is that they are nonpolar. And you remember what I said about nonpolar? They are nonpolar covalent bonds. So things like this, you don't have any areas that are electronegative or electropositive. Those electrons are shared very equally. So therefore, if there are no electrical or electronegativity or electropositivity, there's no point where water can come in and attach to. Hence, you've probably heard the term that oil and water don't mix. Even though water is a universal solvent, it doesn't seem to cling on to anywhere where fats are present. Hence why you'll see a separation. And that has a profound influence on how we can get certain fats to move through our circulatory system. If you were to look at fats, there are fats in your blood. How do we get fats dissolved in the blood? Stay tuned in our endocrine lecture where we talk about how that takes place. The categories, fatty acids, which we'll talk about. These are typically our monomers that we'll be looking at. Triglycerides are our long-term storage of fat. Phospholipids are something that I'll talk about. We have a slide that talks about this. Phospholipids are amphiphilic, meaning they're both polar and nonpolar. They exhibit both properties. Amphi meaning like amphibian, both. And then philic, like philia, like necrophilia, the love of. Steroids are another example that I use. Complex and important, because steroids, which we'll talk about, are very important in our endocrine lecture. So let's take a look at our first example, which we're going to skip to triglycerides. And triglycerides, you'll see on here, again, we don't really need to know about the biochemistry behind this, so you don't really have to remember the structures of these. You don't have to remember how many carbons there are. I want you guys to get the general gist of it, that this is how we store fats. So here are our fatty acid molecules. This one example is palmitic acid. And palmitic acid is a long hydrocarbon chain attached to a carboxyl group. Hence the term why we call it fatty acids. It's an acid because it contains a carboxyl group. Now, the other molecule on here is a glycerol molecule. It is a type of sugar alcohol. And it has 3 attachment points right here where we use C-hydroxyl groups. And in this process, in order to get that fatty acid to bind to that glycerol molecule, we're going to undergo dehydration synthesis. And you guys already mentioned it earlier, is the loss of water. And in this loss of water, we also have to invest energy in it. So this is an endergonic reaction. in order to build that molecule, and I know I had a question here earlier to my left that talked about what type of molecules are you building. This is exactly what I'm talking about. I'm attaching this fatty acid to the glycerol molecule. That's a reaction that involves energy in order to create. And when we invest that energy, we're going to see that a water molecule is lost. Hence why we have dehydration synthesis. So this is what's called, and again, not a term you need to know, but it's called an ester linkage, where we have the hydrogen from a hydroxyl group attached to the hydroxyl group from a carboxylic acid, specifically this fatty acid. And you remove these, the H2O, and you get the water loss, and you get that ester linkage. We repeat that 3 times, and we have a triglyceride, okay? Or otherwise known as a triacylglycerol. You don't really need to know that, but triglyceride is a fat. So if you see this, this is our way of storing fat for us, triglycerides. And if you were to think about breaking this molecule apart, if we're going to break this for energy, imagine breaking bonds here, all these types of bonds. This is going to release a bunch of energy. If you don't believe me, look at fats, look at fuels, look at oils. You burn them and they release a lot of energy. Hence why if we need to store energy, you've had a huge caloric intake and you need to store that energy, you might store some of it as glycogen, but most of it where you're going to end up storing it is in the form of triglycerides, fat deposit, because we've got a lot of energy in those bonds. The next thing we're going to talk about is phospholipids. So I want to take that last concept that we talked about, that triglyceride, and I want to take one of those attachment points where that ester linkage is supposed to happen between the fatty acid and that third point on that glycerol molecule. And instead, I'm not going to attach a fatty acid to it. I'm going to attach a phosphate group. And this phosphate group, I'd like you guys to be familiar with. This phosphate group right here is a very electron-hungry functional group. Why? Because it has 2 things that I talked about, phosphorus and oxygen. Now you got a bunch of double bonded oxygen attached to that phosphorus. That is our phosphate group. And it really longs for electrons. So this makes it very electronegative. And on the other side, as I mentioned earlier, water doesn't have any attachment points. So if you were a water molecule, which side do you think you would be most attracted to? Would you be attracted to the area where you have all these nonpolar covalent bonds, or would you be attracted more to the phosphate group? Phosphate group. So here you see this molecule, the phospholipid, exhibits that property, that amphiphilic. Amphiphilic. You might even see the term amphipathic. 2 terms are interchangeable, where you have a nonpolar side and a polar side. And this is one of, in my opinion, and probably many other opinions in the scientific community, is one of the most important molecules in our, if you believe in evolution, the evolution from unicellular organisms to complex cellular organisms. Because this molecule exhibits that amphiphilic property of having both a polar head and a nonpolar side. This, when you look at these molecules, we're going to look at this on Tuesday. When you dump a bunch of phospholipids into water, okay, and I'm just going to draw a bunch of polar heads like this. You dump enough of it in water, you're going to get the polar sides, and you'll see all my circles are all the polar sides. And all the double fatty acid tails all are hydrophobic. They're going to hide away from water. You're going to get a bunch of water on the outside, and you're going to get all the fatty acid tails that are hydrophobic to gather on the inside. This, you don't need to know this term, it's called a micelle. It's one side where you have an aqueous environment, and on the inside you have the opposite of an aqueous environment is a anti-aqueous environment. Don't quote me on that. I don't know what the real term is. So that is what we have. And if you were to take an even bigger amount of phospholipids, right? If you were to take an even bigger phospholipid, what happens is you have you're going to have phospholipids that are going to create what's called a phospholipid bilayer. I'm just going to draw it like this so that you guys get the point. Where the inside is going to have an aqueous environment. Remember, these are the polar heads where you're going to have water on the inside. You have polar heads on the outside that are going to be exposed to the water on the outside. So water on the inside, water on the outside. And you're going to have this impermeable barrier. And I don't want to say impermeable because it's not impermeable. But you're going to have an area right here that's going to maintain separation from the internal environment and the external environment. This is going to be the major setup of what we're going to be talking about on Tuesday that's going to create the cell. The cellular environment, where we're going to have an intracellular environment and an extracellular environment. And if you look at the creation of cells in the evolution of time, we found that, and this is some of the studies that were done by USC when they were looking at some of the early formed cells. They had like these dust particles that were able to create, and these dust particles had these complex lipids on them that created the separation of the aqueous environment that had toxic stuff in it, had sulfur compounds and all this stuff, and an inside environment that was separated from that could allow stuff to kind of move in and out between these phospholipids, that created this hospitable environment, something where a premature cell could survive on its own, where it keeps all the bad stuff out, keeps all the good stuff on the inside. So that is the whole concept between phospholipids. Let's take a look at our unsaturated fats and saturated fats. Again, not really that important. So I'm okay if you guys don't really get this, but this is really the difference between unsaturated fats and saturated fats. Don't forget, don't remember any of these trans, cis, cis. These are just the configuration, or the molecular configuration of these. It's perfectly okay. But basically, saturated fats are they're linear in nature, and they can stack together and they can get compressed. So you get a lot of fats that are compressed together in a small amount of space. Hence why they can form these solid structures at room temperature. Unsaturated fats have a tendency to be kinked, meaning they are just kinky in nature, and they have a tendency to kind of just float along and never really stack on top of each other. So if you look ever at like peanut oils, you ever look at olive oils, for instance, they're kinked fats that just kind of float around. So they are not as high in caloric density. Steroids. I want to talk about. Steroids are the key component that I was talking about that we're going to be looking at in our endocrine lecture. And the main characteristic of steroids, if you were to look at a steroid, you can tell it's a steroid based on the molecular structure. And here you can see 4 fused rings of carbon rings attached to one another. You'll see some functional groups like a carbonyl group, you'll see a hydroxyl group attached at certain areas along that group. And wherever those functional groups attach will determine the characteristic of that particular steroid. We've got numerous types of steroids that we will be talking about a little bit. If you want to know which ones these are, this one's testosterone. The one on here is dexamethasone, which is a drug that's used for anti-inflammatory. The other one on here is cholesterol. Cholesterol is another important one. Again, you see 4 fused rings attached to a hydrocarbon on there. Something that you'll be able to see in our cellular physiology lecture. You're going to be seeing cholesterol intermixed with a bunch of phospholipids. That's going to give this fluidity. Again, stay tuned on Tuesday. We'll talk more about that. Again, so if you were to see these 4-ring compounds, you can tell they're steroids. And they serve as signaling molecules. As I mentioned earlier, we saw testosterone. Testosterone that's released by the testes has many targets that it can affect. It'll come in, and whenever testosterone cell binds to testosterone, it'll undergo a bunch of changes. It's our way of literally sending a chemical signal to a bunch of other cells. And that's all based, again, on those attachment points. Not something that I would really have you guys memorize per se, but what I'd like you guys to be familiar with is just the inherent structure of those. So what questions do we have about the fats? Shall we move on? Okay, I'll take that as a yes. Proteins. Proteins, if you were to take a look at the monomer of proteins, the single unit of proteins, are those amino acids. So you take that CC, right? And this was our, we had H, 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 and again, you don't need to know the structures of these. We took our ethane, we got rid of that one of the hydrogens, and we added our amine to it, our amine functional group. And in fact, I'm going to get rid of 2 of these, and I'm going to attach a double bond O to it. I have an amine group attached to a carboxylic group attached. That is our amine group, our amino acid. And it's an amino acid because of that, oh, there, I'm missing the other OH. This makes it an acid, the carboxylic acid. So this amino acid is a very versatile amino acid because there's 20 forms that are available on this earth. We have 20 particular amino acids. You see this essential amino acid mnemonic? Yeah, don't worry about these. That's okay. I just want you to just be able to see the, and you can see this on the next slide over. Again, you don't have to memorize these amino acids. I just want to be able to show you that on here we have different characteristics of all these amino acids, of which we have 20. And the main difference between all of these, if you were to take a look at each of these structures right here, these are all similar. Repeating for each single one afterwards. The main difference is the functional group that attaches to that center carbon, that R with the big circle I drew around. This is the question of what attaches to that amino acid. That's going to define what type of amino acid it is. And you can see a bunch on here. You'll see glycine, alanine, serine, threonine, cysteine, valine, leucine. You go through all of these. Tyrosine, aspartic acid, glutamic acid. You'll see a bunch that we do produce. Of which, and again, I'm not going to have you guys really differentiate between the two. We have essential amino acids, which are required for us to be taken in through diet. We have a non-essential amino acids. These are amino acids that we can produce in our human body. So if you think about why certain, when you look at diets and they say get foods that are rich in amino acids, make sure you have a varied diet, it's because you want to make sure that you're getting all of your essential amino acids. Yeah. So now what we're going to do is we're going to take each of those amino acids and we're going to string them together to create a protein. We're going to polymerize it to create a protein. And what we're going to do, let me just show you from our last slide over. Again, we would undergo dehydration synthesis. Again, something you don't need to know. But if I have here's my amine number 1, and then I'm going to have my amino acid number 2, where I have N, etc. And then I have my H. What I'm going to do is I'm going to take these two together. I'm going to remove that, and I'm going to form a bond between this carboxyl carbon and attach that to the other end. And what that's going to do is going to string 2 amino acids together. Now take that image and string a bunch of amino acids all together, like a long Pez string, or just like a pearl necklace of proteins, a pearl necklace of amino acids that are all strung together, and that's exactly what you're going to be able to see. One side is going to be the amino acid end, which you saw here. Here's the amine group. The other end is going to be the carboxylic acid end. And so you get this whole string of what example did I use? Pearl necklace, right? Yeah, I did say pearl necklace. That string that you're going to be able to see here, if I can sort of point that out. 1, 2, 3. You guys get the idea. This is going to string together, and we're going to create a long protein. Now, the thing is that you need to know about proteins. We don't have just one amino acid. We don't have just 2 amino acids. Proteins can be really, really, really big. And I mean, you can string amino acids. I'm talking about hundreds of thousands of amino acids that go on forever. And We make a bunch of proteins. What we're going to be talking about on Tuesday, I believe, we're going to talk about the manufacturing of proteins, how cells produce these proteins. Because these proteins vary in their functions, which we'll talk about in a little bit. What I want to get at first is the difference in the structures of them. And keep in mind that these structures, we start off with a primary structure, a secondary structure, a tertiary structure, and a quaternary structure. These structures build on top of each other. So you got to really just focus and just kind of think 3-4 dimensionally with me. So we take that primary structure and we string just some random, we'll take glycine, alanine, and serine. Glycine, alanine, and serine. We're just going to string a bunch together. That is our primary structure. So if you look at primary structure of a protein, it's literally just a sequence of amino acids strung together, all the way from the very beginning, all the way to the very end. Now take that, and what we're going to look at is the secondary structure. And the secondary structure is looking at the hydrogen bonds that form all along this primary structure. And maybe somewhere on, let's take a look at tyrosine. Oh, there's tyrosine. There's an OH right there. There's another, glutamic acid has another electronegative side on there. Some of these amino acids can now stick together due to hydrogen bonding. So what happens in a secondary structure, all along that primary structure, somewhere those functional groups that attach will be attracted by hydrogen bonding because of electronegativity and electropositivity. When they happen in repeated sequence, you get a series of structures, of which which we'll talk about. An alpha helix is a bunch of amino acids. Again, take that same structure that we looked at, string them all together, and now we get some sides that stick together through hydrogen bonding. And we see a repeated sequence that occurs. And you'll see that it creates a spiral. Let me just take a glance at some of these right here. Secondary structure, if you were to look at the protein being drawn, you can see the secondary structure forming because of these little hydrogen bonds that form. The other thing that you would typically see are pleated sheets, meaning you'll get the primary sequence that goes this way, and then it kind of wraps around and wraps back the other way, and it creates hydrogen bonds that form like rungs on a ladder. And it forms literally like a sheet, a pleated sheet on top of each other. And if you were to take a look at what that looks like, a pleated sheet looks exactly like this. And based on that structure, based on those sequences and the functional groups that attach to those sequences, again, you're going to get another structure. And because those hydrogen bonds are weak, get a bunch of them together and they hold together very strongly. So it's not uncommon when you look at a protein and you look down, you will see these structures. So that's the secondary structure. Again, based on hydrogen bonding. The third, again, now stay with me. Third is protein folding. And based on the regions of this protein that's starting to fold, there'll be some areas that will be hydrophobic and areas that will be hydrophilic. Some areas that will want to stay away from water and areas that will love water and get more exposed to it. There's also going to be these sulfide bridges. If I go back and take a look at any of these that have sulfide bridges, I should see one. Oh, look, methionine has got one. Cysteine has them. You get any of these two that are somewhat close together, and if they come close enough, and you might not see on here, but if they get close enough, they'll bridge together and they'll hold very strongly. So that's the tertiary structure. Let me zoom in on this right here. So take a look at our tertiary structure. Again, if you were to follow the sequence of amino acids, that's your primary. Here you see the hydrogen bonds forming. That creates the alpha helices. Here's our beta pleated sheet. This is our secondary structure. And then these areas right here might be filled with a bunch of hydrophobic amino acids. I'm sorry, hydrophilic amino acids, where they want to get exposed to water. Maybe these areas on the inside are hydrophobic, so they want to stay away from water. So there's a lot that goes into protein folding. Let me get into the last one. I forgot this other one. The quaternary structure. All this one's a little bit simpler. You're taking protein strands that have folded together, and you get another protein strand that comes and folds together, and they'll kind of just stick together like a lock and key. And that's exactly what hemoglobin is. Hemoglobin is made from 4 different protein subunits, and I'm talking about 4 of these structures that stick together and create one functional protein. It's kind of like, is it Transformers? Do Transformers do that? They kind of bind together and create like one super, no, Transformers don't do that. Voltron, do you guys remember Voltron? God, I'm so old.
Power Rangers.
Power Rangers. Power Rangers do that? Yeah, there you go, Power Rangers. So Power Rangers come together and they create some, that's exactly what they do. They form together and they have one function together. And Power Rangers, whatever they did, they were meant to kick ass. That's all they were supposed to do. That's basically what a protein does. A protein does specifically that, and they can be made from multiple structures. What questions do we have about these sequences? I know it's kind of a lot building up on top of each other. Let's take a look at these right here. One thing I want to bring up, is anybody familiar with the protein folding programs out there? There's a, I forgot what institution does it, but I think it was at MIT or one of the other big ones. They have these computers that will take sequences of primary structures. And if you ever look at DNA, and we'll talk about this on Tuesday, you look at DNA encodes for these proteins. So you take these proteins of the primary sequence and you create these long strings, and they can be hundreds of millions of amino acids strung together. The question is, how does that protein fold together, accounting for the secondary structures, the tertiary structures, and the quaternary structures? And there's a lot of computational effort that goes into folding this protein that will determine its inherent shape and determine its inherent function as a molecule. So we take that, and there's no single computer that can do it. So what this program does is, if you install it on your computer, what you're doing is you're donating the computational resources of your computer. It stays on all night, and it runs your processor, and it runs your computer quite hard. And you're contributing the efforts, and it's really cool. They string a bunch of computers together to create this huge supercomputer to calculate the protein folding that occurs. So you can enroll in the program. I don't think you get paid for it, but you contribute quite a bit to science because they've got to figure these things out. If they've mapped the human genome and they want to know ultimately how a protein folds, they have to use the computational efforts of multiple people. And I think as computers get stronger and stronger now, obviously they're making a lot of progress, and they can determine how it folds out. So that is the protein structure. Now that I've mentioned it, the ultimate shape of a protein will determine its function. And you can either have 2 types of protein shapes. Either it's globular, meaning like a globe, like this thing, or you can have a fibrous protein. Fibrous protein you might think of like beef jerky. When you tear them apart, you look at muscle proteins. Those are fibrous proteins. Think of connective, like tendons and ligaments happen to be fibrous. These are more like your connective tissue. These are more of like your globular proteins, things we'll talk about in just a bit. And if you take a look at the globular proteins on here, you can see the areas on here, and the areas in yellow are hydrophilic, meaning they want to be exposed to water. The areas in green have a tendency to hide away from water, so they're hydrophobic. So again, the tertiary structure depends on how this thing folds. You'll see things like an alpha helix right here, secondary structure. In our fibrous protein, you can see 3 different subunits. How do I know? I see 3 different colors. And again, you'll have hydrogen bonding that will attach these structures together to lend itself to the shape of the protein. So the shape of the protein, again, determines its function. Same with this other globular protein. Same thing. You can start to see where different subunits come together. Hydrophobic areas on the inside. Subunits coming together, and then it serves its function. So proteins are really the most diverse and most common type of proteins. The globular proteins we find in almost everything. If you ever do lab work, they're always looking at different proteins, and there's different proteins that they're looking at that will correspond to different functions. You ever look at liver enzymes? Enzymes are proteins. If you look at all the stuff in labs, we'll be able to look at labs and I'll be able to point that out later. Okay, so I kind of use the term enzymes, and that's exactly what I'm getting to, is that certain proteins can become enzymes, right? If you were ever to look at an enzyme, I would say 99% of the time, it's a protein. And I say that other 1% can actually have maybe made from RNA, but really the main idea is that all proteins are not enzymes. So if you were to use that example, enzymes are all proteins, but not all proteins are enzymes. So enzymes are specific. They're a protein with a function. It has one task only, and that's its, and what that task is inherently dependent on what that enzyme is. These enzymes have certain areas to them. If you look at the creation of this protein, we've talked about primary, secondary, tertiary, quaternary structures. There's certain areas based on the way it folds that attracts certain things such as our reactants. And those things that bind to it are called active sites. So if you were to see substrate is just a fancy word for things that bind to it. It's things that bind to something else. And so your substrates in this case would be man and woman that would bind to that enzyme. And that enzyme only binds to whatever it's tuned to bind to. Yes, I'm sorry. It's attracted to it. It literally will fit like a lock and key. So if you look at the shape of man, and this enzyme is supposed to bind to it, it'll be the exact opposite formation of it that allows it to literally bind together. So that's the active site. The enzymes are extremely efficient at what they do. They find a substrate, man binds to it. They find woman, binds to it. And they catalyze a reaction. And what I mean by that, stay tuned, I'll talk about that in a couple slides. We also have the ability to turn off enzymes on and off. If I wanted to do something, turn it on. If I wanted to stop doing it, turn it off. And there's areas on that enzyme that I can do to turn it on and turn it off. So the next thing on here is when we look at enzymes, there's a lot of enzymes. And you can see there's some enzymes on here, but this is not all of them. If you were to ever look at the list of enzymes, we have hundreds of millions of enzymes. Maybe that's an over-exaggeration, but we have lots of enzymes in the human body. And how do we name them? The naming convention is actually fairly simple. If you were to look at every enzyme, the name of that enzyme ends in ACE. So if you were to look at, let's go back to, who are our lactose intolerant people again? Okay, great. So we're going to use that example. Lactose is a combination of glucose and galactose. It's a disaccharide that's bound together that is that milk sugar. People that are lactose intolerant do not have lactase. Lactase is the enzyme that breaks apart lactose. So if you lack that enzyme, maybe you don't lack the enzyme, you might not produce enough of that enzyme to break it down. Therefore, it starts making its way through the gastrointestinal tract. The bacteria that's in our large intestine doesn't react to lactose very well, and it causes flatulence, or it causes stomach ache, or whatever it causes that are typical symptoms of people that are lactose intolerant. That lactase enzyme has one job, and that's to break down lactose into glucose and galactose. Every one of these enzymes have one job. Hydrolase. Its reaction, or what it's responsible is for hydrolysis. Use water to break apart a molecule. That's all it does. For instance, an example of a hydrolase, and these are the classes of enzymes. An example of lipase is to find out a lipid and break it down into a smaller lipid. That's really it. If you look at lactase, that was another form of a hydrolase. It was to turn lactose into glucose and galactose. The other one on there, the exact opposite of hydrolase, is a ligase. is to join two or more chemicals together to undergo an anabolic reaction. The first one I talked about, hydrolase, was a catabolic reaction. So in this case, this allows for the synthesis. If I'm going to turn glucose into glycogen, I'm going to employ some form of a ligase or a polymerase. The other one that I want to look at that'll also be relevant for us is phosphorylase. Phosphorylase, the purpose of a phosphorylase enzyme is to catalyze the transfer of a phosphate ion. That's phosphate, when you see PI, it's phosphate ion, to a substrate. It means add a phosphate ion to that molecule. So once we do that, well, we'll talk about what phosphorylases are for. You'll be able to see that at the end of today's lecture. So like I said, just to recap, enzymes are specific to what they do, and they have active sites that bind to the substrates, and it only binds to that substrate. So like I said, go back to that initial slide that we talked about earlier, when we talked about activation energy. And that activation energy that we talked about was that little push on that roller coaster. It was that match that we needed to set something on fire. It was that little bit of energy that you need to invest to continue that reaction to bring it to its final product. And to use that example earlier, I said man and woman, we needed a little bit of activation energy. And in that activation energy, whatever it is to get them into the right position, to finally bump into each other and produce our product, our baby. Now, enzymes, think about what enzymes do. If they've got an active site, let's take a, let me use an example of a matchmaker. Matchmakers work really well. I'm sure amongst your friends you've got matchmakers that are like, hey, let me meet so-and-so. And they come up and they grab you and they grab the other person and they say, I want you to meet so-and-so. And then they get you two together and then you come together and then you form baby. That's exactly what those matchmakers do. They know the man, they know the woman, they bring them together to create the final product. So in those cases, enzymes reduce the amount of energy that's required. The product of man and woman or reactants stays the same. If we measure the amount of potential energy, that remains the same. If we measure the potential energy in baby, that remains the same. We're not changing the structure of baby. We're not changing the structure of man and woman. All we're doing is we're helping that process take place a little bit. They're matchmaking to bring them together. So in that case, an enzyme is programmed to attach to a man, attach to a woman, and help the reaction come together and form the product. Okay? They are meant exactly for that. So in that case, we reduce the activation energy. There's no change in the potential energy. So I'm going to draw that change in potential energy here, which we mentioned is negative delta G. There's no change in this. This remains the same. It's just the activation energy that comes in. So this again, to use another example, if you had a stack of papers, I couldn't even really think of this would be like a flamethrower. I can't even really use that as an example. You'll learn this about me. I'll try and like play around with analogies and I'll just be like, no, that doesn't work, just kidding. In this case, that's where we reduce the amount of effort or energy that's required to start the reaction. If I were to take this whole beaker filled with reactants and products, and I throw, and let's say this reaction starts to slow down, right? Because I needed a good amount of energy to keep this going. If I threw some enzyme, if I threw a matchmaker in there, that one's going to come in and grab A and grab B together, put them together, and form more of the product. It speeds up the reaction because you use less energy. And that I'm referring to as activation energy. What questions do we have about that? Because I'm going to expand on this. Good? All right. So now we have to think about that enzyme. And what we're going to be looking at is ways to regulate the activity of that enzyme. Maybe that matchmaker works way too fast. Slow down. I don't want that much baby. I need more men and women. I don't need too much baby. In fact, you've created too many babies, turn that enzyme off. Okay? So in this case, what we're going to be looking at, we're going to be looking at competitive inhibition, we're going to be looking at allosteric regulation. And there's two different ways of looking at where we can compete with. Now think of the example of competitive inhibition that I was talking about earlier. I've got an enzyme that comes in. Here's our matchmaker, and we have the substrate, which is man, and we can use the example of woman later. Substrate comes in. This is one of our reactants that comes in, and it's supposed to bind to the active site on that enzyme. So this is our active site. A competitive inhibitor comes in and binds to the active site because the structure of the inhibitor mimics that of the substrate. So as I mentioned earlier, lock and key, this competitive inhibitor kind of looks like that key, and it can fit into that area. And what that ends up doing, it literally will block that active site. So think of that example that I mentioned earlier. You have that matchmaker that comes in and say, hey, I really want you to be so-and-so. You're going to come up to that man, but now you're going to get this competitive inhibitor that comes in here, and I've used this term before, comes in and just kind of cock blocks right there, right in the center. Just says, no, don't do that, don't do that. In fact, I'm just going to get in the way. That's exactly what a competitive inhibitor does. Now, a reversible competitive inhibitor still competes for the active site, but it's reversible. Pretty self-explanatory. So it may bind sometimes, and in that case, if it does bind, it prevents a substrate from binding. But if we were to use this example earlier, a bunch of men, women, baby, and I threw that enzyme in there, the amount of competition that takes place is dependent on how much reversible competitive inhibitor I have. So if I toss in like one cock blocker in there, it's just going to block one enzyme. If I toss a bunch in there, it's going to come in and start competing. You got more of these competing for the active site than you do have your reactants. Does that make sense? So think about it this way. If I have more substrate is likely to bind to an enzyme than a reversible competitive inhibitor. If I add more reversible competitive inhibitor in there, you have more of these that compete than substrates. That's how you can regulate, and that is something that we typically see of ways to compete for substrates, to modulate how much product we form, because it'll take that substrate, it'll undergo its conformational changes, and then it'll produce that final product. But get stuff that comes in and competes for that site, and you can limit the rate of reaction. Now, while we've been talking about the active site, in terms of competition for the active site, we have to talk about another site. And this is what's called the allosteric site. It's another site on the enzyme that is not the active site. So if this is your active site, there's an allosteric site that can bind to it. And in this case, this is an allosteric inhibitor. An allosteric inhibitor comes in, and when it binds to that allosteric site, it creates an entire change to the conformational change of that protein. These can hydrogen bind to this area and cause a change in the shape of the protein, or change the shape of the enzyme. And when you change the shape of the enzyme, you can change the shape of the active site. That's why proteins, when you look at proteins, any minor change, any tweaks to the protein can really cause dysfunction of that enzyme. It could potentially be that people that are lactose intolerant may have some mutation on there that changes the active site. It's now distorted so it can't bind to, so that lactose can't bind to that active site. You still have the enzyme, it's just misshapen. Okay. Give you a relevant example. Sickle cell anemia. People that have sickle cell anemia, the shape of the hemoglobin, actually it's the shape of the red blood cell, but the shape of the hemoglobin may not lend itself to be able to attach to oxygen. Again, that might make more sense when we go in later. But the main idea is attach an allosteric inhibitor and you distort the active site, substrate can't bind to it. Now the converse of that is an allosteric activation. Take that same allosteric site, attach an allosteric activator, and that now activates the enzyme site. So that active site can now bind to the substrate. So in other words, in order for this enzyme to operate, it needs its partner. It needs this allosteric activator that binds to the allosteric site to cause a conformational change to the active site that allows it to bind to the substrate. I know I strung a lot of words there, but that's basically the summary of it. So inhibitor and activator, allosteric on the allosteric site. All right, so looking at the enzyme phosphorylation, we're going to take a look at, and I'm not really going to even go into this, but enzyme phosphorylation. There was a phosphorylase enzyme that comes in that phosphorylates that enzyme. Typically what you would see that would activate an enzyme is typically a phosphate ion. That's something that you'll commonly see. In order to turn on this enzyme, I need a phosphorylase to come in here and attach to that enzyme. And yes, I did use that example of an enzyme attaching to another enzyme. There's a lot of enzymatic reactions that require numerous enzymes to activate it. So it's like a matchmaker. Take that matchmaker enzyme, and that needs another enzyme to come in and turn on that matchmaker. You can string a bunch of enzymes together to create that one function. So you take that phosphate ion, attach it to that enzyme, and then it turns it on. Typically, you would see this. This is an example of an enzyme that's involved in cellular division, something that we'll talk about on Tuesday. Cellular division takes place, and there's something that will turn this back on. And they'll do it so that it can stop that cell from continually dividing. And we find that if there's any mutations to this enzyme, we may find that that enzyme might not turn off, or that enzyme might not be turned on anymore to stop cellular division from taking place. So any mutations to an enzyme can potentially cause a cell to continually divide if it affects specifically this enzyme. I don't want to lose you guys too much on that, so I'm going to skip this altogether. Any way you look at it, this is just a way of turning it on. Other ways to modulate an enzyme from taking place. Doesn't always require an inhibitor or an activator. Other things that we can talk about. Temperature. If I get an enzyme and I turn up the temperature a little bit, think of that matchmaker. Now it's working up fast because it's got a lot of kinetic energy from all the heat. Now that matchmaker works really fast. If I cool it down, if I bring the temperature down, we reduce the kinetic energy, and now that matchmaker moves very slowly. You modulate the rate of the reaction due to temperature. You want to get that sugar to dissolve fast in water, heat it up. If you want to slow it down, cool it down quite a bit. pH. If I start introducing hydrogen ions in the bath, hydrogen ions can come in and start binding to the enzyme. And that could start to denature some of the proteins. Think about this. When you eat beef jerky, or if you're whatever, tofu jerky, or whatever, anything protein you take in, when you're eating it, it goes into your stomach that's filled with acids. Part of that breaking down, you start to see the hydrogen ions that start to attack it and start to change the shape of the protein and denaturing it. That's a term that I'm talking about, denaturing a protein. By denaturing it, you start to render that protein to become ineffective. Okay? So take an enzyme, put it in an acidic bath, and that can start to change the effectiveness of that enzyme. Substrate concentration. Matchmaker will not work very well if there's not enough of your reactants in place. So if I have less reactants, those enzymes, they're just looking around trying to find man and woman. They can't find it. You start dumping a bunch of reactants in place, and now they start working faster. Again, Le Chatelier's principle. The rate of reaction depends on the rate of your reactants. Increase the number of these, you produce a lot more of these. Reduce the amount of these, and you produce less of your product. So with that in place, you now have the idea, and the last couple slides just I want to finish up with, is this lends itself to the whole negative feedback system. I want you guys to take what you learned on Tuesday. in terms of the feedback system and apply it to this, is that the rate slows as the product accumulates. Once, take that bath again earlier, take that enzyme that's in there. If I'm trying to create a balance of, and I don't want too much man and woman, I don't want too much baby, I want just the reaction to take place. I don't want to have it too fast or too slow. If it happens too fast, then I get too many babies that are produced, too many products that are produced, less of my reactants. Therefore, the reaction slows down. Okay? If it's too slow, right? If it's too slow, I don't have enough baby, I have more of my reactants, man and woman, therefore the enzymatic reaction speeds up. And start introducing all the inhibitors and activators that can be introduced into the system to regulate the rate of how much we produce something. So that is the whole system behind negative feedback. We want it to operate in a certain way, and when there's numerous physiological mechanisms that we introduce in the way to slow the rate down or speed it up if we need it to. The last thing I'm going to kind of brush through, which is still of utmost importance, are nucleic acids. I've got 1, 2, 3 slides left. Nucleic acids are the fourth macromolecule that I want to get into. And this is the structure of DNA and RNA. Something, again, we'll talk about on Monday that we'll get into some detail about, enough for you that you should be able to answer some questions on the quiz, and then real technically not really worry about it afterwards, because this goes into a lot more detail that I don't need you guys to get into. Again, a lot of this is biology. DNA is called deoxyribonucleic acid. RNA is called ribonucleic acid. So these are the structures, and as you know, DNA is the backbone for our basically Human Genome Project. It's the sequence for what makes us who we are. All the components that we're ever made of is all stored in our DNA. And the way it's stored, if we take a look at the monomers are the nucleotides. These are nucleic acids that are formed. And we have, if we look at the monomer, the nucleic acid is comprised of 3 parts: a nitrogenous base, a pentose sugar, pentose meaning 5, and it's 5-carbon sugar, and a phosphate group. And you can see where these are. These green things, phosphate ions. Pentose sugar, take a look at that pentagram right there. That's our pentose sugar. This right here is our nitrogenous base. The nitrogenous base, we have 4 different nitrogenous bases. Actually, technically we have 5, but for DNA, we have 4. You don't need to know pyrimidines, you don't need to know purines, you don't need to know the names of these. Don't worry about that. We do have thymine and cytosine, adenine and guanine. These are single ring bases and double ring bases. At no point I'm going to ask you which one. Thymine, is the thymine a single ring base, or is guanine a double ring base? It's not really important to physiology. But the thing that we do need to understand is that adenine only binds to thymine. A double ring only binds to a single. Adenine to thymine and guanine to cytosine. There's no rules outside of that. They only bind to A to T and G to C. So if I were to take a look at this side of the sequence right here, A, G, T, C, and you'll see that the way DNA forms, phosphate, pentose, phosphate, pentose, phosphate, pentose, it strings together. And you have your nitrogenous bases attached together. I'm sorry, let me restate that. Nitrogenous bases attached to the pentose sugar. It creates this sequence of AGTC, AGTC, a bunch of nitrogenous bases that create this entire long sequence. Did you guys ever watch Jurassic Park? Or Jurassic Park, and they showed you, if you ever watched the portion where they're looking at the DNA sequence of dinosaurs, and it was a bunch of these ATGCAG, and it was like this really long string. It's exactly what it is. If you were to zoom in, if I were to zoom in on your DNA and look at the sequence for your eye color, I would see that it's going to be this hundreds of thousands, millions of gajillions of these nitrogenous bases that will give us the sequence of your DNA. These are genes that encode specifically for that. And if you were to look at what's called the complementary sequence, adenine, if I only had one side of it, adenine would bind to thymine, guanine would bind to cytosine, thymine would bind to adenine, cytosine would bind to guanine. So when we learn cellular division later on, you'll see how DNA will come in and it'll get spliced. The thing is about DNA, I kind of forgot to mention, DNA is a double helix. You probably have already seen this before. It forms this look to it. It's a double helix. And these right here, you would see the nitrogenous bases on the inside. And they're held together by hydrogen bonds. You see these two right here? These are the hydrogen bonds that form, that create this long ladder of DNA. And this area to this area could be the sequence for your hair color, could be the sequence for skin color. It gives you the pretty much everything in there. There's all the different genes that we have that encode for these. This could encode for certain proteins. This could encode for certain enzymes, tons of it. So that ladder that's formed together is the hydrogenous bases. RNA, ribonucleic acid, is a single strand of nucleic acids. And we'll talk about that on Tuesday. The three types of RNA, again, I'm not going to really require this out of you guys, but RNA, we've got three types of RNA. One that's going to be partly involved, part of the construction of a ribosome, which we'll talk about Tuesday. tRNA is what's called an amino acid transfer. We go into this. This is actually one of your first lab assignments that we're going to end up doing is how do we sequence out the construction of a protein from DNA. We're going to go from DNA to RNA to the protein itself. Messenger RNA, again, we'll talk about on Tuesday. Now, take one specific nucleic acid, one of these DNA RNA structures. We're going to take one nucleic acid where we had a, this was specifically an adenosine, this was an adenine nitrogenous base, and we attach, here's our, there it is, adenine, this is our adenine. We attach that pentose sugar, which we call ribose, and instead of a single phosphate group that you saw earlier, that same green thing you saw here, instead of a single phosphate group, we string 3 phosphate groups together. The name of this structure is called adenosine triphosphate, and it's an important molecule because this forms the energy currency in our body. It is what holds a lot of energy in it. If you look at energy transfer, all of this is all contained within these phosphate groups. Each of these bonds of phosphate groups are very strong. They've got a lot of energy that's contained. And if you take this ATP molecule and we break off one of those phosphate ions, that's going to release energy. Remember, we're breaking this apart. It's a catabolic reaction. We take ATP, break off a phosphate group, and we get ADP, which adenine diphosphate, plus your phosphate ion, plus, don't forget, this is an exergonic reaction, energy. Tons of energy that can be coupled to endergonic reactions. Things such as building your protein. So if you're building that protein, you're going to be doing that in the lab assignment. It's a really easy lab assignment. If you're building a protein, it's going to require energy, and that energy comes in the form of ATP that's required. So with that, that breaks off our phosphate group. If you were to ever think about, has anybody ever watched The Matrix before? Yeah, you remember The Matrix? There was a group of, and again, I'm going to digress a little because I know it's like 9:40 and I know you guys are eager to go. In The Matrix, there was this a bunch of series of animations that were done before called the Animatrix, and they do the whole thing that talks about why the machines were taking humans and harnessing energy from them. There's a portion where Morpheus says the humans were nothing more than a battery, and he's talking about all that bioenergy that's available. Where does that bioenergy come from? Is it in the form of heat? No, it's actually in the form of ATP. So in the early in the Animatrix, they talked about the not ATP, but they talked about the energy that they were able to harness. And this is basically bioavailable energy. If you had a whole cup of ATP, that is energy that can be used to convert into readily form of energy. So that is ATP. I want you guys to be familiar with this. Be familiar. I want you guys to make sure you review. You've got all of this weekend. I'll make sure that I'll get out the links to you guys. So make sure by tomorrow, send me your email addresses. I'll get the list of email addresses. I'll get you the links so that you guys can get the access to the Google Drive. I'll make sure I go in and also find out the statuses of your enrollments. If you see anything funky on your enrollment, let me know and I can forward that over to the registrar's office. Have a good weekend. Good luck studying. I will see you on Tuesday.